All right, so we're starting a new unit today, which is Unit 2. And Unit 2 has 4 lessons to it. There is Nervous System Part 1, which is what we're doing today. And then there will be a Nervous System Part 2, which deals more with the anatomy of the brain and the spinal cord. And today is more about the physiology of how a neuron works, for example. And then we will have special senses. So we'll look at the eyes, we'll look at the ears, and the 5 senses that we have. And the last lesson for the unit will be on the digestive system. So let's get started. And I'm just going to do a little recap of everything that you are supposed to know for this lesson. All right, so we have nervous tissue, which is separated into basically 2 categories. Okay, we have the neurons themselves. I should probably call this nervous system part 1. Okay, so there are the neurons themselves, and then there are the glial cells. Glial cells. Give me a second. Hold on, someone request transcript here. Let me turn on the transcript. There we go. And then there are the glial cells, which are the supporting cells of the nervous system. Okay, they are also called neuroglia. Okay, so these are supporting cells of the neuron. Does anybody have any examples of glial cells? Ones that we talked about in the lecture.
Schwann cell.
Yeah, that's right. So I'll list some examples here. Thank you. That's a Schwann cell. Okay, which is important for forming myelin in the peripheral nervous system. Anyone have another type of glial cells?
Yes, oligodendrocyte.
Yeah, oligodendrocytes. Thank you. Okay, and this is responsible for forming myelin. And if you don't know what myelin is, I'll explain in a second. In the CNS, central nervous system. Okay, and I'll explain those things again. Right? And then there are other glial cells that was explained in the lecture, such as astrocyte. Astrocyte. These form the blood-brain barrier. There's also the ependymal cells. Ependymal cells, which are responsible for forming the CSF, cerebral spinal fluid. Okay, so those are the supporting cells. They provide protection for the neurons. They provide nutritional support for the neurons. Okay, so as you know, neurons, they don't really divide, but the glial cells, they are the ones that can divide, and brain cancers arise from glial cells and not the neurons themselves. Okay. So let's just look at a neuron, a typical neuron. You should be drawing this with me, right? And taking down the notes, hopefully. And so a typical neuron looks something like this. All right, and there's going to be like a long projection from the body, like that. So these things that are projecting from the cell body, that's the receiving end of the neuron. And we call them, actually, anybody know what they're called?
Dendrites.
Yeah, they are dendrites. They are dendrites, okay? And this is responsible for receiving signals from another cell. Usually it is another neuron, but it doesn't have to be. It could be a sensory receptor, right, at the beginning of the circuit. And we can talk more about that if you forgot what a sensory receptor is. Okay, so that is a dendrite. And there are two types of signals in general. We have signals that are inhibitory, okay, like these red arrows. Inhibitory signals. They will tell the neuron to stop firing, to stop passing on the signal. Okay? And sometimes you want to do that, right? Sometimes you want to suppress certain electrical signals, right? For example, if you're studying in the cafeteria and there are some background noises, people chatting, and you just really want to focus on your work, so you would kind of zone out the background noise, right? So that would be like an example of where inhibitory signal is used. Another example is, if you think about your arm, in order to flex your elbow like this, right, you would have to inhibit the tricep, the muscle at the back, right? And while you contract the bicep. If you want to extend your elbow, then you want to inhibit the muscle in the bicep while you contract the tricep. So there are various reasons of why we need to inhibit signals in the nervous system. And of course, the opposite of inhibitory signals are these, anybody know the name? The opposite of inhibitory?
Yes, excitatory.
Thank you. Excitatory. Excitatory signals. Okay, and at any given time, a neuron can be receiving a mix of these signals. And at the end, the neuron would add up all these signal. It's called signal summation. And whichever one is stronger, that's going to be the end result. So if there are more excitatory signals than inhibitory, then the neuron will transmit the signal. On the other hand, if there are more inhibitory signals than excitatory, then the neuron will suppress the transmission. Okay, so you would have your nucleus here in the middle and all the organelles that you're supposed to have in a regular cell, in the mitochondria, the Golgi body, and all that stuff. Okay, now this here is a particularly long extension, and we call that the axon. Right, the function of the axon is to transmit the signal down, right? And so the signal initiate from the cell body, and it goes all the way down the axon. Okay, and when the signal gets to the very end, this location is called the axon terminal, right? Which is responsible for relaying signals to another cell. And usually that's another neuron, but it doesn't have to be. It could be a muscle cell, could be an endocrine gland, could be an organ. That would be the axon terminal. Now, in order for the signal to not get weakened as it's passed down the axon, many axons actually have these insulation. These are like fatty substance that wraps around the axon. Okay, and these are called myelin. Okay, and if we are talking about the central nervous system, which is the brain and the spinal cord, then the oligodendrocyte would be responsible for forming the myelin. If this is a neuron in the PNS, your peripheral nervous system, then the myelin would be formed by the Schwann cells. Okay, but in any case, the purpose of the myelin is to speed up conduction. It allows the signal to pass really quickly, and you will not lose the strength of that signal. Now, you do not wrap the entire thing in myelin. You need to leave a little bit of gaps, and these gaps, yeah, it's fatty substance. That's right, Ahmed. These gaps, they are called nodes of Ranvier. Okay, and that's where you will have ion exchange. Okay? If you watch the whole recording, you would know about the action potential, which is just ion exchange between sodium and potassium, and those exchange occurs at the node of Ranvier. Right, and what happens is the signal will basically jump from node to node, and it will be able to reach the axon terminal rather quickly. Okay, so there you have it. That's your typical neuron. Does anybody have any question for me? We good to go? All right. Now, not all the neurons are going to look like this. Some neurons are what we call bipolar. Okay, and a bipolar neuron, they're going to have like 2 axons instead of 1. Okay, so that would be like a bipolar neuron. Okay, so cell body right here, and then it would have one end over here. It will receive a signal, and then signal goes to the body, and then it goes to the other side. All right. So there you have it, the structure of a neuron. Now I want to go through the flow of information again. Flow of information. I believe I did that already in the recordings, but I realized many of you have not watched the recordings, so I'll do this again. Flow of information talks about how the information is transmitted within the nervous system. So in order to trigger some kind of pathway, right, you probably need to have some kind of stimulus, okay, a stimulation, right, a change in the environment, for example. Yeah, I'll answer your question in a bit, Ahmed. It's coming up, okay? Just hang in there. Oh, no worries. Don't have to be sorry. Over here, let's say there's a hand, your hand. All right, and you touch something sharp, like a needle or something. Okay, there's a needle, okay, that pokes you. All right, so how do you detect that, right? How do you feel that poke? Well, it turns out throughout your skin, you have all these different types of receptors. Okay, so in general, we call them sensory receptor. Sensory receptor. Okay, and you just have to trust me, they kind of look like this. Okay, kind of look like this. All right, so the sensory receptor is responsible for detecting a change of some sort. All right, you have different types of sensory receptors. You have sensory receptors in your eye, in your eyes that detect changes in light intensity. You have sensory receptors in the skin that detects changes in temperature. In this case, we would have a sensory receptor that detects changes in pressure, right? To feel the poke of the needle. Right? So that's the sensory receptor. And what's going to happen is once it detects the poke, right, the change in pressure, the sensory receptors will then send a signal to a sensory neuron. Okay, so a sensory neuron is a bipolar neuron. Okay, there's the body, and then there's one axon, right? And there's another axon. Okay, so this is a sensory neuron, right? The job of the sensory neuron is to take sensory information to the central nervous system. Okay, so sensory information could be basically anything that you detect with your senses. Okay, so it could be light information, could be temperature, could be texture that you feel with the hand, like you touch the table, it feels rough, right? If you touch something sharp, right, that's a texture. That's an example of sensory information. Smell, taste, all these things are sensory information, and in order for these information to go from the sensory organs to your brain, or spinal cord in some cases, we would need to have the sensory neuron. Okay, now there is not just a single sensory neuron, because like that's super delicate, and if you break one, then you basically can't feel, right? So there are like a bunch of them. Okay, a bunch of them. I'm just gonna draw like 3 so that we don't get like super confused. So there is a bunch of them, and this whole thing forms what we call the sensory neural sensory forms the afferent pathway. Okay, afferent pathway, which basically takes information from the PNS, the peripheral nervous system, anything that's not part of the brain and the spinal cord, towards the CNS. Okay, and the afferent pathway is made up of sensory neurons. Okay, gonna pause for a second, see if anyone want to ask anything. All right, well then.
Professor,
Yeah, go ahead.
the afferent pathway, is it like capillaries and arteries, like blood tracks connecting brain to the body?
No. This is has nothing to do with the vessels. Okay, this is completely just talking about the nervous tissue. Okay, so it has nothing to do with the arteries and stuff like that.
Thank you.
Okay, so then the information is going to travel through the sensory neurons, and we are going to have another type of neurons in the central nervous system. What do we call this neuron that is within the central nervous system?
Interneurons.
That's right, thank you. This is the CNS, and this is the PNS. Okay, so CNS is brain plus spinal cord. Okay, so within the spinal cord or the brain, we have the interneuron. And the job of the interneuron is basically processing the sensory information. Okay, so process the sensory information and determine the appropriate motor output. Okay, in other words, how are you going to respond to it, right? When you touch something sharp, then the reflex is to, let your finger, lift your hand and put your finger out of the way of the needle, right? So that's a motor output. Okay, and all that happens within either the brain or the spinal cord. In this case, it's the spinal cord that does the reflex for you. We call that a spinal reflex. Some things involves, more complicated decision, then you would have to involve the brain. Okay, so once you figure out what to do, then you have to send the information back out. And so there's going to be another set of neurons that will carry the information all the way back to the hand. Okay, like that. There we go. And that is what we call the motor neuron. Motor neuron. Okay, so it takes motor information towards the effector. Okay, so the effector could be a muscle, a gland, or an organ. Okay? Don't confuse this effector with the effector that we learned in the previous unit. Okay, it's not related. So, like I said, it's not just one of them. You have a bunch of them. Okay, and it forms the efferent pathway. Okay, so it carries information from CNS towards your PNS, and then ultimately it goes to your effector. Okay, and it's made up of motor neuron. Okay. Any questions? Okay, so all these like a bundle of neurons, we actually have a name for them. Okay, so let me just, oh, what happened here? Everything it's gone. Let me go back here. There we go. A bundle of axon, right? A bundle of axon is called, right? So a bundle means a group of them, right? Like this. So a bundle of axon is called, I should say a bundle of axons in the PNS, okay, is called a nerve. Okay. So this here, this efferent pathway, like physically you would have a nerve there. And you can also have a bundle of axon in the brain or spinal cord, but we would call it a different name. Right? It's called a tract. Okay. So if you take a look at the brain, right, here's the brain, and then here's your spinal cord. We're looking at it from the back or something. Okay, and the spinal cord, you might or might not know, it becomes like a ponytail at the bottom, right? Okay, so this is brain. And this here is the spinal cord. And together it makes up your CNS, central nervous system. So coming out directly from your brain, you would have a bunch of nerves. Okay, so these ones, they are called cranial nerves. How many pairs of cranial nerves do we have?
12.
That's right, 12 pairs. 12 pairs, yeah. Okay, and if it's coming out from your spinal cord, then we call them spinal nerves. Okay, and that we have 31 pairs. Okay. So this nerve, right, we'll learn more next week, but this nerve is basically going to be the bundle of axon, right? The sensory nerve and the motor neurons, they're actually going to be grouped together in a single bundle. Like if you, I don't know if anybody still have a telephone at home, but then if you look at the telephone cable that you plug in, if you cut that open, inside there are two lines, one for sending transmission out and one for receiving. And that's exactly how it looks like in the body as well. Okay, so both of these things are going to be grouped together in a single bundle to form the nerve. All right, more on that next week. It will definitely make more sense by then. Okay. So moving on, just like summarize more. We have the nervous system again, nervous system. Which I've been telling you, it's separated into two branches, right? And we have the central nervous system. Okay, which once again, just to reiterate, is brain plus spinal cord. Okay, that's it. That's where integration occurs, processing of information, right? And then everything else, right? The spinal nerves, the cranial nerves, everything that's outside of the brain, outside of the spinal cord, we call it the peripheral nervous system. Okay, the PNS. So these spinal nerves, these cranial nerves, they are part of the PNS because they are not within the brain or the spinal cord. And the PNS is functionally divided into two divisions. Okay. We have the autonomic nervous system, which is called ANS. And the other division is somatic nervous system, which we call the SNS. Okay. So basically, the ANS is responsible for everything that's autonomic, everything that you don't think about, right? So like beating of the heart, right? Automatically breathing, right? You don't even have to think about breathing for it to occur, right? Digestion of your food, right? All that stuff is going to be governed under the ANS. And anything that involves voluntary control, that would fall under the SNS, right? The somatic nervous system. So like to talk, right? You would need the SNS. To move around, right? That's SNS. Now, under the ANS, it further divides into two functional division. We have the sympathetic nervous system, which is called sympathetic system. And the opposite of sympathetic is parasympathetic. Okay. So these two divisions, they are antagonistic to each other. They target the same organs in many instances, but the effects on the target would be opposite. So for example, they both target the heart, but the sympathetic system will cause the heart rate to go up, whereas the parasympathetic system is going to tell the heart rate to come down. In general, when is the sympathetic system most active? Both system is going to be working at all times, but during certain periods, one is going to dominate over the other. So when will the sympathetic system be dominating?
Like a fear when we.
Yeah, exactly. Thank you. So when you are in danger, right? Fear. When you are stressed out, right? Or when you're excited, right? Like on a roller coaster ride, or you feel very pumped up, right? From winning a race or something like that, a marathon, for example. So the sympathetic system was going to be most active during those times. We call it the fight or flight, right? Response. Okay. When you're in danger, you either fight the thing or you run from it, right? Flight. So that's the sympathetic system. And on the other hand, the parasympathetic system is going to be most active when you are just, resting. Okay, when you're relaxing. Right, so this is the time to, for example, digest your food. Digest your food. Right? Go to sleep. All right, so they are always opposite to each other. Okay. So let's just try to go through this situation again, right? The pricking of the finger with a needle, and we're gonna see which system does what. Okay. So the finger, right? Fingers, we have complete voluntary control over it. And so it's not surprising, it shouldn't be surprising that it's going to be starting at the somatic nervous system. Okay, so you're going to detect the prick. Pricking your finger. Okay, so that's going to send a signal, right, through the sensory neuron all the way to your nervous system, right? Sorry, wrong path. Oopsie. It's going to send a signal all the way to your central nervous system. In there, that's where you will have the interneuron. Okay. And then what will the interneuron do? Well, the interneuron will send the appropriate motor response back out into your PNS, right? And then it will tell you to remove the finger. Now, it's not always like this. I'll give you another example. Okay. I'm just going to erase this. Let's say you are like walking into walking from a dark sorry, let me just rephrase that. Like, let's say you wake up in the middle of the night and then someone switch on the light. Okay, someone switch on the light. Then what happens to your pupil? Does your pupil get big or does your pupil get small?
Small.
Yeah, because it's bright light, right? Exactly. Right. So do you think that starts from the somatic nervous system or the autonomic nervous system to detect that? Sorry, go ahead.
Autonomic.
Yeah, exactly. Because we cannot control our pupil, right? Like, I mean, I certainly cannot. I don't know about you. So what happens is that's gonna have a signal here, and then it's gonna go all the way to the central nervous system, and then again we will have our interneuron, and then the appropriate motor response will go out. And do you think it's going to be the sympathetic system or the parasympathetic system if the light goes on? Which one causes your pupil to go small? Yeah, good guess. Actually, is the parasympathetic system that makes it go small. Because when you're in danger, your pupil dilates. You want to see better. You want more light to come in, right? Okay. So constriction is actually the other way around. Okay. I mean, you don't really have to know exactly that, but I'm just showing you that the pathway starts here and then it can go this way. Let's just do one more, right? Okay. Let's say you are driving and then you're going a little bit too fast and then you see a cop car or something like that. Okay. And so immediately, right, that is a visual input, right? So from the ANS and go to the brain, and then you process that information with the interneuron. And then what are you gonna do? Well, you gotta slow down. So you're gonna go to your SNS, and then it will tell your legs to press on the brakes. But at the same time, right? It will go to your ANS, and specifically it will go to your sympathetic system, because then you're afraid to get a ticket, right? So your heart goes boom, 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 right? Increase your heart rate. So sometimes it sends to multiple division to have multiple effects. It doesn't have to be a linear circuit. Is that okay? Already. That's basically a lot of the information already. Let's do some polls and see where we're at. Over here. Sorry, my poll button is being blocked. Okay, there we go. Go ahead. All right, thank you for participating. Let's take a look. Basically anything that's not brain or spinal cord, you can check it. All right, because cranial nerve is outside of the brain. Spinal cord is part of CNS, sympathetic system, spinal nerve, parasympathetic system. So anything but spinal cord and brain would be the PNS. Okay, let's try another one. An impulse from one nerve cell is communicated to another via the I don't think I've mentioned it today yet. It's certainly in the lecture recordings, but I'm just gonna tell you the answer. There is the neuron, right? Oops. There's the neuron, and that's the axon terminal, and then you're gonna have another neuron, right? Like that. So if I zoom in into that location, you will see that they actually do not touch. They just come in close proximity. So this location is called a synapse. Okay, so that's the correct answer. Synapse. All right. More on synapse later. Okay, this one. Go ahead. So I can see the class is split between the two. I mean, it's one of them, right? And the CNS is the oligodendrocyte. Okay, the Schwann cell is for the PNS. Okay, for the PNS. All right, that is basically it. Okay, so the study guide, this was already filled in during the lecture. Okay, and we just drew it again, so there's no need to go through it. I'm gonna talk about the action potential now. All right, the action potential. So let's go through it. I mean, once again, like the detail explanation, you should watch the recordings. But I'm just gonna go through like I guess not a summary, but then like I'm gonna go through it, okay? But then it's not gonna have as much detail as the recordings. Anyway, do ask questions if I'm not making sense. The quiz, right? The quiz is next Tuesday for this lesson. Sorry about the confusion. I know the day was wrong initially, but it's next Tuesday. So here is your neuron, and we know the signal it's gonna pass down the axon, right? Okay, I'm not drawing the myelin just to keep it simple. So we're gonna learn about something called the action potential. Action potential, which is basically the mechanism for signal transmission down the axon of a neuron. Okay. So again, I invite you to draw along with me. I strongly believe when you draw things out, it would help you understand better. Okay, so like if this is your paper, right? Like don't draw a tiny diagram like that. It's gonna be really crowded and really hard to see, right? Okay, like go like this. Okay, that's the axon that I'm drawing. So that's what we will be doing. We will zoom in on this location, and that's the axon. So in the axon, right? This is the outside. We call it the extracellular fluid, the outside. And this would be the cytoplasm, which is the inside of your neuron. Along the cell membrane of the neuron, you will have various membrane proteins. And the ones that we are interested in are these purple ones that I'm drawing. You would have quite a few of them. And they are called sodium potassium pump. Using ATP, the sodium potassium pump is going to pump sodium out of the cell. And it's going to pump 3 sodium out in exchange for what?
Potassium.
How many?
2.
That's right, 2 potassium in exchange for 2 potassium. Okay, so that's the job of the sodium potassium pump. It keeps on pumping 3 sodium out in exchange for 2 potassium in. And so after a while, you will end up having high sodium on the outside. Okay, you will have high sodium on the outside. And you will have low sodium on the inside. On the other hand, you would have low potassium on the outside and high potassium on the inside. Okay. Since we are pumping more positive out than we are bringing in, over time, the outside will be more positive. Compared to the inside. Okay. So this difference in charge distribution, we call that the membrane potential. Right? And right now the neuron is not doing anything, is not transmitting a signal. So the resting potential is approximately negative 70 millivolts. Okay. So the function of the sodium potassium pump is to create the resting potential. Okay. Now, if we go back to our study guide here, right, that's what we're at. Okay, on the y-axis, we have the membrane potential measured in millivolts. And over here on the x-axis, we have time, right? Okay. Volt is a measurement of electrical potential. Like if you think about a battery, like a AA battery or something, and then you look at the voltage, it's 1.5 volt. And so it means between the positive and the negative terminal, there is a charge difference of 1.5, right? So what in the context of what we're talking about, we have positive on the outside and negative on the inside, and the difference between the outside and the inside is minus 70 millivolts. Okay, so there are 1,000 millivolts in volts, right? So that's what a potential is. So number one here, right? This we call the resting potential. Resting potential. And the number, the magic number is minus 70 millivolts. So let's take a look at the key events that's taking place here. Actually nothing much is going on. Okay, so no signal. We can say the neuron is at rest. No impulse. Impulse means signal. Okay, and at this point, we have on the inside of the neuron, we have low sodium and high potassium on the inside. Right? We can say in the cytoplasm. Okay. It's always going to be about the inside unless otherwise specified. Okay, so that's basically what's happening. Okay. I'm going to go back to our picture. In addition to all these sodium potassium pump, we will also have these channels. Okay, so you will have sodium channels that are closed right now. These, think of them as like little doors. Okay, they're closed. That would be open. That would be closed. Okay, think of it again. It's like doors. Okay, so sodium channels, they are closed right now, and then you will also have potassium channels, which are also closed. Okay. So in addition to the sodium potassium pump, there are also sodium channels and potassium channels. Okay. Now these channels are controlled by voltage. Okay. And each of them will have a range of voltage where they would open. And beyond those range, they would close. For example, the sodium channels. They will open at minus 60 millivolts. And then they will close at, does anybody remember what number did they close at?
Yes. Theory.
Yeah, that's right. Positive 30 millivolts. Okay. If you read the textbook or like some online stuff, it might be like, they might say 33 or something. I just round it to 30 so it's easier for you to remember. Whereas the potassium channels, they will open at 30. And then they will close at minus 90. Okay, so each of them has a designated range where the channels will open. And if you're outside of that range, they will close. So currently we are at minus 70, which means it's outside of the sodium channels opening range and it's outside of the potassium channels opening range. Okay, that's why everything is closed right now. You might be thinking, but minus 70 is within 30 and negative 90. Well, you have to open them first, right, before you close them, right? You cannot do it in the reverse order. Okay, so you must reach 30 first, you know, before closing. Okay. Does anyone have any question before I move on? Okay. So, what's going to be happening? Well, let's think about this. We have high sodium on the outside and low sodium on the inside. When given the chance, do you expect the sodium to come in or do you expect the sodium to go out? Let's do a poll on this one. Just want to see if you guys are with me. If you think, the question is this: when you open the sodium channels, do you think the sodium is going to come in or go out? If you think it's going to come in, please choose A. If you think it's going to go out, please choose B. Not as definitive as I liked it to be. The correct answer is in. Okay, and the reason is because things naturally go from high concentration to low concentration, right? So we have high sodium on the outside and low sodium on the inside. When you open these sodium channels, the sodium would come in. Okay. Similarly, when you open the potassium channels, the potassium would leave the cell because we have high potassium on the inside and low potassium on the outside. Okay, so that's something to keep in mind. Now, how do we get things started? Right? How are we going to trigger the signal? Well, the sensory receptors that we had at the beginning, once they detected a change in the surrounding, they will initiate a signal. Okay, so there is going to be some kind of stimulus that's going to be arriving. Okay, a stimulus that's will be coming in from the cell body, and that will slowly push the membrane potential towards positive. So let's go back to our graph here. Number 2, you see there's pointing at this point here. Okay, that is when the stimulus arrives. Stimulus just means some kind of trigger, okay? And at this point, we are still sitting at minus 70. But what's happening is the stimulus will push the membrane potential towards the positive direction. Okay. Now some signal will not be strong enough, so it will go up a little bit and then will just come back down. Okay. But other times the signal will be strong enough to push all the way towards -60. Now, if you have watched the recording, you would know that -60 is a significant number because -60 is what we call the threshold potential. Threshold potential. And if you have been listening to me, then you will recognize -60 is the triggering point for the opening of the sodium channels. Okay, so the threshold potential is -60 millivolts. And what happens is the sodium channel open, right? Which allows sodium to move. Which way again? In or out? In your mind, you should think about it. Open the sodium channel. Do they come in or do they go out? That's right. They will move in. Move into the cell. Okay. I mean, it's kind of obvious on the graph, but then I want you to think about it as well. When the sodium is coming into the cell, do you think the cell will become more positive or more negative on the inside? Right? Look at our picture. Originally, it's more negative, right? But with all the sodium coming in, when you open the sodium channels, is that going to make it more positive or more negative? Okay, so both Tina and Amit said positive, and they are correct, right? And that's why in our graph, on a graph, you see that this is keep on going up, right? In fact, you are going to cross 0, and you are going to become positive, okay? And so number 4 here, this whole section, this is called depolarization. Okay, depolarization. The color don't mean anything. Okay, I'm just like choosing colors as I please. Okay, so depolarization occurs from -60 millivolts to positive 30 millivolts. Okay, so what's happening here? Well, the opening of the sodium channels will cause a sodium influx. Okay, which then what the sodium influx does is it will reverse the membrane potential. The membrane potential was negative initially, but now you open all these sodium channels, you add all the sodium come in, it will flip, it will reverse, it will become more positive on the inside. Okay. And this will continue to happen until 30. Okay, so what happens at 30? Do I have another pointer? I do not. I'll explain it over here. So at 30 millivolts, what's going to happen is the sodium channels close. And the potassium channel will open. Right? That's the opening voltage for the potassium, if you remember. Okay, so this is depolarization, the rising phase of the graph right here. We can label it like that. Depolarization. Okay. All right, now that's 1:30. Let's take a quick break. We'll resume in about 5 minutes. Okay. See you in a bit. All right, let's keep going. So, what's the situation so far? All right, let's draw a new picture to see what's going on. All right. Let me get the right thickness here. Okay. So what's had been happening so far is we have all the sodium coming in. Okay, I'll represent them with red dots. Okay, and the outside would have fewer. Okay. Now remember, the axon is really, really long. All right. So when the stimulus arrived here, we are only causing the sodium to be coming in for the beginning section. The rest of the neuron is still in its initial condition, right? It's still at the resting condition. All right. So let's put some markers here to indicate the sodium level. So over here, we have low sodium because they all came in, and therefore it's high on the inside. But further down the axon, we will still have high sodium on the outside and low sodium on the inside, because those sodium channels, they are still close. They have not reached the triggering points yet. Okay. If I measure the potential difference down here, it would still be at -70, right? And that's not the opening voltage just yet. All right. So with all the sodium coming in, what number are we sitting at right now? It's not -70 for sure. But what's the new number that we're going to put right here? That's right. It is going to be +30. Okay, +30 millivolts. So by the time we reach the peak of the graph, by the time we reach the +30, all the sodium channels would close once again. Now I'm ignoring the sodium potassium pumps because otherwise it would be too crowded. All right. And then what happens is the number 30 is the triggering point for opening the potassium channels. Okay, so the potassium channels will now open, right? Further down the axon, though, they will still be closed. So what's the potassium situation here? Just before these things opens, is the potassium high or low on the inside? Before anything happens, like we just reached 30, right? It's still high because that's the initial condition, right? If you go up here again, look, it was high before. It's still high because they haven't gone anywhere yet, right? And we would have low potassium. But as soon as we open these potassium channels, then the potassium will start to leave, right? So we have all these potassium very concentrated on the inside. They will all start to leave, okay? And as the potassium is leaving, you will once again start to decrease your membrane potential because you're losing positive charges, right? Okay. And these potassium really want to leave, okay, for two reasons. Number one, there is a concentration gradient, right? There's a concentration gradient. Let me write this down for you. The potassium efflux is driven by, number one, concentration gradient. In other words, they like to go from high to low. And number two, there is electrostatic repulsion. Okay, there's so many positive charges on the inside, and they just want to push each other away. Sodium can go out because the channels are closed, so the potassium are the only ones that are leaving. So these two factors combined together will cause a lot of K plus, a lot of the potassium ions to leave the cell. All right. And so here we can see on our graph, right, number 5, this is coming back down, caused by the potassium efflux, and we call this repolarization. I cannot write sideways. Sorry, I'm just gonna write like this. Repolarization. Okay, you're recreating that negative charge on the inside. So that's number 5. I'll write here again. Repolarization. Okay, so that would be going from positive 30 all the way to minus 90. Okay, all the way down, right? Because there's so many of them leaving, driven by the two forces, right? So here it's potassium efflux. Okay. Membrane potential returns to negative once again. Okay. All right, so let's go back up here once again. I'm gonna put a few more markers here just to remind you. We started with the low sodium and high potassium. Okay. What about at the peak here? What was it again for the sodium? Is it high or low? That's right, both high. High sodium and high potassium. High sodium because of the sodium influx, and then high potassium because they didn't go anywhere yet. Okay. And then repolarization, that is the potassium efflux. Okay, and at the very bottom here, right? What about the sodium now? Is the sodium high or low at this point? Yeah, sodium is high, right? Because they all came in, but they didn't really leave yet. So that's high sodium. And all the potassium left. Well, not all of it, but like a lot of them left. So we have low potassium. Okay. So at number 6, we have a name for this. This is called the hyperpolarization. Okay. Hyperpolarization. It's too negative. That's what it means. Minus 90. Okay. So 2 things is wrong here. Number 1, ion distribution is opposite to the resting state. Number 2, membrane potential is too negative. Okay. So these 2 things makes it impossible for the neuron to transmit another signal immediately. Okay, so we must reset the neuron back to the starting state before it can pass on another signal, right? And so I want to ask you, what's responsible for redistributing the ion and resetting it back to minus 70? What is that protein that is going to be responsible for doing that? Mavis says the pump. That is correct. What's the name of the pump though?
Sodium and calcium pump.
Sodium and potassium, that's right.
Sorry, potassium, yeah.
Yeah, no problem. Sodium potassium pump will reset it. Will reset these conditions. Are we together? No, it's a lot of information, but hopefully it makes sense to you. All right, so there's another thing that we should talk about over here, right? How does the signal move forward? Well, like I said, after the sodium comes in, after the sodium influx, we have a lot of positive charges here. The potassium get pushed down, no problem, through their open channels, but the sodium has nowhere to go, okay? It cannot go back out because the channels are shut, but there are so much positive charges on the inside that they will get pushed away from each other. So there's only one direction to go in this diagram. And what direction would that be? I'm talking about sodium here, guys. Okay. Further down, it's correct. Okay, it's going to go further down because we have high here and we have low here. So some of the sodium, it's going to go forward. Okay, and remember, these sodium are positive charge. And so as they go forward, this number is going to change. It's going to start to go up, right? We are at -70, and then with more sodium coming in, it might go to -65, and then -63, and eventually it will hit -60, which would cause the sodium channels to open, and then this next section will experience the exact same thing that just happened in the previous section. And then more sodium will move forward, and that's how the signal gets passed down. All right, so let me try to show this to you. You don't have to draw this next part, you can just watch, okay? So there we go. We have a very long axon right here. In the beginning, they are all sitting at -70. -70, -70, -70, okay? And so what happens is a stimulus arrives, a trigger, some kind of trigger that will push the initial section over the threshold, right? Over the threshold. So I'm going to draw another neuron here. It's the same neuron, okay? But like maybe a millisecond later. All right. So what happens is this is going to get pushed over the threshold. It's going to become -60. Sodium channels open. All the sodium comes in. And next thing you know, this section becomes +30, while the rest of it is still sitting at -70, right? So this section is currently experiencing the action potential. What all that means, like what it basically means is a temporary flip in the membrane potential. Okay. So now we are at the peak of the graph, right? If you remember our graph, it goes up and comes down, right? We are at the peak right here, the 30 plus for this section anyways. Then what happens is some of the sodium gets pushed forward, okay? And if enough of them get pushed forward, the next section will edge towards the threshold potential, right? And so once you cross the threshold of -60, then the next section will become +30. The rest of it will still be -70. But what happens to the original first section? Well, that was at the peak at the 30, right? And so after the sodium channel closes, the potassium ones will open, causing all the potassium to leave the cell and bring it all the way down to what number?
-9.
That's correct, -90, right? So this is in the hyperpolarized state. It's not ready to transmit another signal. We have to wait for the sodium potassium pump to reset it, okay? And now this section is the action potential. All right. Let's take a look at what happens a moment later. A moment later, the -90 would have been reset back to -70 thanks to the sodium potassium pump. This section would now plunge into the hyperpolarization voltage, while the next section will get pushed up to the positive 30. Right? And the remaining of the axon will still be minus 70. Right? And so on and so forth, right? So if you visualize this, I have no way of showing you this with an animation. I couldn't find one, okay? But if you visualize it, there's this long axon, right? And then at the beginning, right, the first part lights up, right? Ding, creates the plus 30, which will then trigger the next section to light up. So like ding, 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 right? It just goes all the way until the end. But the key thing is only one section goes ding at a time, right? Only one section is going to be experiencing the action potential at a time, while the rest of it will still be at rest. And then the previous section will be busy resetting itself back to minus 70. So there you have it. That is the action potential. Does everybody understand? Anyone want to ask any question? Anything that keeps you up at night? All right, all right. Cool. I just have a random question for you guys. I don't think you guys had a choice. You had to take this course online, right? But I was just wondering, if you had a choice, would you still, having gone through what you have gone through, would you still want to take this online, or would you want to take this like in person? Okay. I'm just gonna do a poll and see what you guys think. I'm just curious, okay? Go ahead. Interesting, interesting. Half. Although we have a small size here, we actually have 40 people in the class. I don't know where the rest of the 26 people are, but based on the results that you guys gave me, it's half. Interesting. Yeah, it's a lot to do online, right? Like I'm thinking I probably have to teach this thing again next semester in an online environment. I'm thinking to switch out the recordings and just do everything live. What do you guys think about that? Do you think the recording is too much to do on your own, and you rather, come here twice a week to listen to me and instead? Any opinion?
No. For me, that's very good.
Oh, you like the recordings?
Yeah, I like the recording, and we do later on the question, solve the question.
Oh, okay. Because I guess the recording gives you flexibility, right? Like you can do it whenever you want, right? Okay, interesting. Yeah, I have to, wrestle with that one. But later when you guys do the course evaluation, please tell me about these things, right? Because there's no way for me to know what works and what does not know, right? Like, I mean, it's too bad not able to see you guys in person and have these conversations. But please do let me know. I would love to hear from you. Okay. Yeah, I know it's hard to study and have kids at the same time, right? Totally get it. My kids also makes it very hard for me to work when they're at home as well. Tomorrow is PA day, so I'm gonna have to wrestle with that. Anyways, so that's that. That is the action potential. I think we covered everything. We can totally fill this in again, just for good measures, and then we'll talk about the synaptic communication. And from there, I think we will call it a day. All right. When a neuron is at rest, the concentration of I'm just choosing a color. The concentration of potassium is high, and the concentration of sodium is low in the cytoplasm of the neuron. Okay. The resting potential is created by the sodium potassium pump. The pump works by pumping sodium out of the cell and potassium into the cell. This causes the inside of the cell to be more negative than the outside. The resting potential of a neuron has a voltage of minus 70. When a signal arrives, it can cause the membrane potential to rise above minus 60, which is called the threshold potential. Crossing the threshold potential triggers gated sodium channels to open. The opening of the gated sodium channels allows sodium to move, and you should know that by now, into the cell. The influx of sodium causes the inside of the cell to become more positive than the outside, resulting in a reversal of membrane potential. This temporary reversal of membrane potential is called an action potential. Okay, and I just want to quickly remind you the influx of sodium is what causes the depolarization. The reversal of membrane potential causes the gated sodium channels to close and the gated potassium channels to open. The opening of the potassium channels will allow potassium to move out of the cell. And so we're at the very peak now, and we're going to start to come down, right? Causing the membrane potential to become negative once again. Okay, so this is what we call repolarization. At this point, the membrane potential is approximately minus 90 millivolts, and the concentration of sodium is high, while the concentration of potassium is low in the cytoplasm. The membrane potential and ion distribution will be restored by the sodium-potassium pump. Okay. Now I have another thing that it's I forgot to mention in the tutorial. Each channel will enter a refractory period. After closing. Okay. So the refractory period is a period where the channel will not be responsive. Okay, so it opened and then it closes, and then it will enter the refractory period where it's like a time of non-responsiveness. It must reset itself before it's going to respond again. Now that is very important to have the refractory period. Let's go back to the graph and see why. Okay, just take a look at the graph here. Sodium channels, if you remember, it opens at minus 60 and close at 30, right? Okay, so look, we cross the threshold, the sodium channels open, and then the sodium channel closes. But on the way down, due to the efflux of potassium, we actually go past 60 again, right? But the sodium channels, because they're in the refractory period, they will not open even though we hit minus 60, right? You see that? And that's important because if you open them again, then it's just going to go up and down and fluctuate the signal instead of getting the signal going forward. Okay, so that's why it's important to have the refractory period. Just for the record, the refractory period is a period that the channels enter after they close, and they will not open no matter what trigger you give them. Okay, that's it. Let's go and do our synaptic transmission. All right, so we have our neuron here, and then we have another one here. Okay, and then that's our synapse. So here we go. And we're going to zoom in here. This is called a presynaptic cell. Right? And then over here, we're going to have our postsynaptic cell. Okay, so presynaptic cell is the cell that's carrying the signal, and then postsynaptic cell is the one that's going to be receiving it, right? So in the axon terminal of the presynaptic cell, we are going to have these vesicles. And inside the vesicles, we will have these things, these molecules, which are called neurotransmitters. Okay, NT for short. Okay, neurotransmitters. So they are chemical messenger for synaptic communication. All right. Now, it's important to know that well, a couple of things to know. Okay, I'm not going to write all of it down because it's already in the lecture notes. I'm just going to refresh your memory about it. Okay, so a couple of things to know about neurotransmitters. Number one, they can be excitatory. Remember, like the first thing I told you today, over here, right? Like here, some signals are excitatory, some signals are inhibitory. So excitatory signals would be passed on using excitatory neurotransmitters. Okay, inhibitory signals will be passed on using inhibitory neurotransmitters. Okay, so the neurotransmitters, depending on the type, there are many different types, right? Give me some examples. Do you guys remember any from the lectures or any that you know? Any neurotransmitters that you know of? You guys haven't got that far in the in the recordings. What's the happy hormone people say? Right, you eat a cookie, makes you feel good. What's being released in your brain? Starts with a D. Yes, dopamine. Okay, dopamine. Right, there is like serotonin. Right, GABA, acetylcholine. Right, all these things are neurotransmitters. Okay, so number one, the first thing you have to know is, depending on which type of neurotransmitter you're talking about, sometimes they are excitatory, sometimes they are inhibitory. Okay, sometimes it can be both. Okay, depending on which region of the brain you find them in. Okay, so dopamine, for example, in certain part of the brain, in the reward system pathway, it's a positive signal. Okay, makes you feel good, right? When you do something rewarding. Okay, like eating a cookie feels good. Okay, running 5K makes you feel good, right? So these are positive reinforcement, right? So that's excitatory for dopamine. However, dopamine in another part of the brain called the substantia nigra, and you can see this in the lecture note, is responsible for suppressing unwanted motion when you move. Okay, so that is inhibitory signal, right? So it's same a neurotransmitter, opposite function, depending on which region of the brain you find them in. That's number one, first thing you have to know. Okay, number two is that in the actual brain, it's not just one-to-one like this. Okay, a lot of times you would have a single neuron, but then there are like so many other neurons in the vicinity. Okay, so like many other ones here. Okay, so I'm just gonna draw a few more just to show you. You don't have to draw it. Okay, I'll erase them afterwards. So when you release the neurotransmitter into the synaptic cleft here, right, all the neurons in the immediate area will be exposed to the neurotransmitters. However, in order for you to respond to it, you must have the right receptor, right? So I'm using color code here. So we have the blue neurotransmitters, and only the neurons with the blue receptors will be able to respond to it, right? So if there's another neuron here, and this one has like a purple receptor, even though it's being exposed to the blue receptor, it would do absolutely nothing, okay? So to use proper terminology, let's say these blue things are dopamine, right? Okay, then you would have to have a dopaminergic receptor on the neuron in order for it to respond to it. Okay, so that's that. Now, what's going on? How does the signal go across? Well, the action potential that we describe will arrive at the axon terminal. Located on the axon terminal are actually calcium channels. Okay, so that's a new thing, calcium channel. We did not see any calcium channel along the axon, but they are at the axon terminal. When the action potential arrives, the calcium channel will open. Okay, the opening of the calcium channel will cause the calcium to move into the cell. Okay, and calcium is important because they will push these vesicles towards the edge of the cell. Okay, and then when they get to the edge, what happens is they will basically release the dopamine, okay, or the whatever the neurotransmitter is. Do you understand? Okay, so action potential comes, calcium channel opens, calcium will push the vesicles towards the edge. You will have exocytosis, if you remember that from last semester, and you will get all these neurotransmitters out into the synaptic cleft. So remember, we were talking about the sodium and the potassium channels, and I told you they are controlled by voltage, right? So it will only open when you hit a particular voltage. Well, these channels, they are actually also sodium channels, but they are controlled by chemicals. Okay, so we call them chemically gated channels as opposed to voltage gated channels, right? And in order for this to open, you would have to have the dopamine bind to it. Once the dopamine binds to it, the channel will open, right? And this is a sodium channel. And what happens is the sodium would come in, right? If enough sodium comes in, it will then flip the membrane potential to positive, and that's how the lightning bolt begins, right? The stimulus that I was talking about, right? You have to have enough trigger to push the membrane potential towards the -60 at the beginning of the neuron, right? Before you can trigger the signal down into the axon. Okay. Now afterwards, we have to clear the synaptic cleft. We have to get rid of all these neurotransmitters, because if they stay there, you would be transmitting the same signal over and over again. So to avoid that, we have to clear it up. Two ways we can clear it up. We can absorb them back into vesicles. We can use it for later. Or sometimes we would have enzymes that would come in and break them down, right? Either way, it doesn't really matter. The key thing is we have to remove them so that we're not going to have sustained signaling. Okay, so that's important. In the lecture, I talked about Parkinson's disease, which is a low dopamine level. We talked about serotonin connected to clinical depression. You should review those slides. There are some questions in the quiz that relates to those concepts. Okay, let's just finish up with the application question here. Adenosine acts as a neurotransmitter. When sufficient level of adenosine builds up, it slows neuronal activities and makes a person feel tired and sleepy. Caffeine is a stimulant and its effects is related to adenosine. Based on the chemical structure shown, explain how caffeine makes you feel energized. Okay, so let's take a look at this. Adenosine, as in like adenosine triphosphate adenosine, right? You've heard that before, right? Like ATP. Okay, so as you metabolize ATP, it becomes ADP plus P. And what you might not know is ADP can actually be broken down further to form AMP plus P. Okay, so then you build up adenosine when you're using up a lot of ATPs, and hence you feel tired. So let's take a look at this situation. Okay, so here is our neuron. I'm just gonna draw a neuron, and then I'm gonna put a receptor on it. Okay, I'm gonna put a receptor. Okay, and the receptor might look like this. Okay, I'm just gonna do it like that. Okay, so this is an adenosine receptor. Okay, so what they're saying is, when you build up adenosine, it will slow neuronal activities. Okay, so here is an adenosine triangle binds to it, right? And then it will it will slow down the neuron. Okay, and then you will feel tired. And then the more you have, the more tired you will feel, and then you go to sleep or whatever, right? So now caffeine. If you look at the structure of caffeine and the structure of adenosine, what do you notice, huh? You notice anything special about them? Just gotta blow my nose. Give me 1 second. Sorry about that. Do you see any resemblance between the caffeine and the adenosine? I hope so. You can see that this part here is very similar to that, right? Okay, so when you drink caffeine and you flood your system with it, and caffeine can actually go into cross your blood-brain barrier and go to your brain. So this is your adenosine. Okay, and the caffeine looks very similar. Okay, it also has that little triangle part, for example. So the caffeine will bind to the adenosine receptors and compete for them, right? So now you have less available receptors for the adenosine, right? So even though you might have a lot of adenosine building up, but they won't be able to bind to your neuron, and therefore you're not gonna feel sleepy and tired. Yeah, in a way, it's a competitive inhibitor. We could say, okay, because it competes for the same binding slot, right, Mavis? All right. So here is something that we should think about. Your brain has high. Oh, it's too thick. Has high plasticity. No, that's not how you spell it. Plasticity. Okay. What that means is, it's not a static organ, right? It changes over time. It changes in response to the environment. Okay. And so a lot of what the brain tries to do is it always try to regain some kind of equilibrium. Always try to regain some kind of balance, okay, that it had before. So in this case, when you drink a lot of coffee, a lot of caffeine, it doesn't have to be coffee, it could be tea, it could be those monsters people drink, caffeinated beverage. When you do that, then all the receptors are kind of like, overwhelmed, right? Flooded, right? And so your brain will try to recalibrate itself to the initial state by putting more receptors on it. Okay, by putting more receptors onto it, right? Just like before, because before there were a lot of available receptors, but now all these receptors are kind of flooded by the caffeine. So to regain balance, it will put more receptors on it. Does that make sense? You guys with me before I tell you the next next part of the story? Do we all understand why the brain is putting more receptors on it in response to the caffeine that you're drinking? Okay. So how many of you drink caffeine, like coffee? Do you guys need a little pick me up in the morning before you start your day? Yeah. For people who drink coffee, right, caffeine of any form, Do you feel that you need to drink more over time in order to feel the effect? Right, and tea could be, yeah. So, you know, when I was in university, right, that's when I first started drinking coffee. First year university, right? I bought a small double. Never drank coffee before before that point. Got a small double from Tim Horton, and I drank it. And my God, I felt so energized. I said, "What is this stuff?" Right? And then you know, even after the test, I had an evening test that night, I remember. And then after the test, I was still so awake, and then I couldn't really sleep at night. And then I started drinking. But then after a while, that small double is no longer good enough. I need to up my dose, so to speak, right? Start to go for medium, and then so on and so forth. And I continue to drink coffee for until after university when I go to work. And then I had to drink like a whole mug in the morning. And that doesn't even do anything for me anymore. However, if I don't drink it, then I won't be able to function. In fact, when I don't drink the coffee, I get these headaches, and I find it really hard to concentrate. I don't know if any of you had that experience. And so, you know, these are what people call withdrawal symptoms. Okay. And caffeine is like a mild version of that. It's much more severe for people who are using, say, opioids, for example, right? Or pain medications. If you just stop it all of a sudden, right, cold turkey, so to speak, right, you have this very intense withdrawal symptoms, and it's very difficult to cope, right? So what happens is this, okay? Remember, the brain is trying to rebalance by putting on more receptors, right? And that's why the same caffeine level is not going to reach the desired effect as it once did, because now all these extra receptors are available for the binding of adenosine, you see? Okay, so in order to flush those out, you might have to drink, you know, a more coffee, right? Like a grande or something, right? Much bigger. And now you have to block those receptors. Make sense? And your brain will still try to counteract that by putting even more receptors on it. Okay, so there's going to be a point where, you know, it's saturated completely, and you are no longer going to be able to feel as much of an effect. Okay, in fact, if you stop drinking all of a sudden, right, you take away all these receptors, you take away all these caffeines, then all of a sudden you have all these extra receptors that weren't available before, and your adenosine can like totally bind to all of these and makes you extra sluggish, extra tired, right? And it gives you all these, like, you know, withdrawal symptoms, the headaches, right? The sensitivity to light, and so on and so forth. Okay, so that's basically what's happening. Now, how do you get off coffee? I eventually did. Okay, like I stopped drinking coffee about 10 years ago, and I actually had to taper myself off. Okay, wean myself off. Right. I cut it down by. I was able to cut it down by 50% initially. Right? So instead of drinking like a full mug, right, I start by drinking just 50, half a cup. And what happens is I was able to cope with that, and then I slowly reduced the dose, right? So then your brain would start to take off some of these receptors that I put in there, and eventually, you know, I was able to get off it, right? So for anybody who's interested about quitting, that's what you have to do. But you know, like I said, right, caffeine is it's a really mild thing, and you know, there's nothing essentially bad about drinking coffee. So most people don't pay any attention to it. But this could be applied to things like pain medications, right? You might or might not know there's an opioid crisis going on, right? For the past couple years already. And it's these people who are taking the drug medication. Initially, it would do wonders for them. You might even have heard of like Oxycontin, right? Which was a very powerful opioid that was used to treat chronic pain because it was advertised as non-addictive. But what happens is it essentially triggers the brain to do what I described to you, right? When you take a little bit of the pill, it works at the beginning, but then your brain try to recalibrate itself, try to reestablish the equilibrium, and you end up having to take more and more and more. And eventually what happens is your body becomes dependent on it, right? And it becomes really difficult to come off it. So that's that. Anyways, sorry for going on a tangent. But any questions you have for me for the first one? Fantastic. Number 2, GABA. GABA is a neurotransmitter that causes Cl- to enter the neuron or K+ to leave the neuron. Based on this information, predict the effects of GABA on neurons with GABA receptor. Okay, so let's take a look at our situation here. This is our, axon. And normally we have negative on the inside, right? Negative on the inside. And then this is positive on the outside. Okay, and then in order to trigger a signal, you would need to reach a threshold of minus 60 millivolts for our sodium channels, correct? So in the presence of GABA, which is the neurotransmitter, right, we have chlorine coming in. Okay, so imagine for a second that the chlorine is coming in and the potassium is leaving. What will that do to your resting potential? Originally, we are sitting at minus 70, okay? But in the presence of GABA, these chlorines are coming in and the potassiums are leaving. Does that cause your resting potential to be less negative or more negative?
Yeah, more negative.
Exactly, because we all negative coming in, right? The chlorine is going to push it even further down. So if you go back to our graph here, you will see that in the presence of GABA, instead of starting at minus 70, you might be down here. You might be further down over here. So that will make it harder to reach the threshold potential, okay? Because now you are hyperpolarized. It will be more difficult to hit the threshold potential, okay? So if you inject GABA into like your arm, for example, right, you will experience muscle weakness because it's so much harder to reach the threshold necessary to cause the muscle to contract, for example. So that's that. Let's just finish off with two more poll questions. This one has to do with Prozac. I mean, I didn't review it with you, but let's just see if you can answer it. Okay, so for those of you who are not sure, serotonin, if you don't have enough of it, it will cause clinical depression. Okay, so just to quickly refresh your memory, this is the synapse that we were talking about, and you will release the serotonin into the gap, and then it would do its job. Okay, and I told you if after the neurotransmitter does their job, you must either break it down or you must take them back up and store them as vesicles, right? You don't want them to hang around the cleft for too long because otherwise you'll be repeating the same signal over and over again, right? Except in this case, because clinical depression is caused by low serotonin, okay, you actually want the serotonin to hang around in the synaptic cleft for longer than they normally would, okay? Again, the problem is you don't have enough serotonin, so you want them to hang around longer and exert their positive effect, okay? And that's what the drug Prozac does. Prozac is an antidepressant, and it will block the reuptake of serotonin from synapse. Okay, if you block the reuptake, you let them stay there longer, and they will be able to compensate for the low level of serotonin. Okay, because you don't have enough of it, right? So you make use of what you have. Okay, you let them stay there for longer. Okay, this one is difficult. It will be on your test quiz. Okay, so please try that. All right, I have answers all over the place. Let's just talk about this. Hypokalemia, that is hypo is too low. Emia is in the blood. So low potassium in the blood can result in hyperpolarization. Okay, so hyperpolarization means that your starting potential is down here. Okay, so which of the following will happen? Some sodium channels will spontaneously open. That is not true. Okay, you are like way more negative than you were supposed to be, right? The threshold is all the way up there, so they're not going to spontaneously open. Okay, resting potential is more negative than they should be. A greater than normal stimulus is required to trigger the action potential. That is true, right? Because you have to go all the way up there as opposed to just this little bit, right? And that's the correct answer. Uncontrolled muscle twitching, that's not true. That would be for hyperkalemia, okay? If you're starting closer to the threshold, then some of the sodium channels will spontaneously open and it will cause your muscle to twitch a little bit. And the threshold does not change. Threshold is always going to be minus 60. Okay. So that's it. Okay. Well, a lot of things to study for. And I hope you'll be okay. I'll see you all next Thursday for the lecture 6. Okay. Have a good one. Bye.